What is up, guys? I'm back with another video, and today we're going to be looking at the cell cycle checkpoints, specifically looking at the checkpoint going from G1 to S. We're going to be looking at all of the various kind of complex subunits and different substrates, such as P53, retinoblastoma protein, the E2F complex, and all of this stuff that can get really confusing, and all of it plays a role in basically controlling going from the G1 phase of the cell cycle into the S phase. And this is important because when you look at the point of regulating this part of the cell cycle, you have to understand that something like DNA damage or cell damage in general, you do not want that cell to proceed into the S phase where DNA replication will eventually occur, and then going all the way eventually into mitosis where the cell will basically duplicate itself. Because if you have a damaged cell and this happens, you can imagine that this is how various cancers can happen, mutations, problems with various cells in your bodies and in different organs. So let's get right into this. I want to really simplify this process into being able to answer all of the questions that they're going to ask you on the step one if they were to ask about cyclins and cyclin-dependent kinases and whatnot. Because for me, I get really confused when they talk about all of the different names, the different letters. So I'm going to just focus on what are the high yield important things to know and simplify this using clever mnemonics so that you won't have to think twice when it comes to test day. So let's get right into this. At the very bottom here, I've written out everything that we're going to talk about. So you already know all the molecules that are going to come into play when we're going from the G1 phase of the cell cycle into S. But before we go into that, I'm going to draw a circle here, and I'm just going to remind you of the different parts of the cell cycle. So remember that right at the beginning, there's something called the G1 phase, where a cell is going to make sure that all of its organelles are in check, that everything is in play before it enters into the S phase. And the S phase is just going to be where DNA is replicated. Well, it needs to have replicated DNA because when the cell divides, of course, you're going to need two sets of the DNA, and that makes sense. So after the replication phase, or the S phase, we go into what's called G2. To put this really simple, G2 is just going to do one final check before we go into the M phase, which is just mitosis. And you remember from high school cell biology, all of the parts of mitosis, usually the step doesn't ask it as much, except for knowing some of the different drugs, but we'll cover that in a different video. This particular video is focusing on going from the G1 phase into the S phase. There's a block here, and this block is called the restriction point. Now, this restriction point is where all of these molecules come into play. For example, say we have a cell, and this is DNA. I'm making this real simple. And there's some damage. There's a damage right here, okay? This is going to be detected by this system using these four kind of clusters of substrates and different proteins and whatnot. And this is going to stop. Our mission is to stop this cell from going into the S phase because we do not want to replicate DNA that is damaged or mutated or any sort of something like that. Okay, so let me show you how this happens. This is how I started out. Step one, almost look at this like a story. What happens first of all is you have DNA damage. Well, what is going to detect DNA damage? What's going to detect DNA damage is P53. That alone has never been hard for me to remember because almost everybody can remember that P53 is a tumor suppressor gene. What does that mean? It's going to suppress the formation of a tumor. And you know that a tumor is just a cluster of cells that won't stop producing. Basically, it's producing too many cells. It's basically bypassing these checkpoints, and so a tumor forms because too many cells are being produced in that local area of the tumor. So P53, if you can remember that this is a tumor suppressor, it will help you to remember what the function of this of P53 is going to do in this pathway. So P53 detects that there's DNA damage over here in the drawing that I drew. What is it going to do? When it detects this, it's going to activate P21. Okay, so that's the first thing you need to know. P53 realizes there's DNA damage, and it's going to activate P21. So now P21 is what's called as a cyclin-dependent kinase inhibitor. a cyclin-dependent kinase inhibitor. So let me explain that by going on to our next little group of molecules. So we covered P53 and P21. Now we're moving on into cyclin-dependent kinases and cyclins. So I'm going to just draw CDK right here. CDK stands for cyclin-dependent kinase. We'll just look at the word. A cyclin-dependent kinase, clearly it's dependent on cyclin, so that tells you it won't work without cyclins, and it's a kinase. And remember from chemistry in college that kinases phosphorylate things. And when you phosphorylate things, things are either activated or deactivated, and it depends on the pathway, because there's tons of phosphorylation and dephosphorylation occurring all over the body. In the case of CDK, when cyclins are around, the cyclin will basically bind to the CDK, and now you have this complex, and it's called the CDK cyclin complex. And there's various names for this, but you just need to know that when cyclins are around, the CDK is now active. That's easy to remember. Let me remind you. Cyclin-dependent kinase depends on cyclins. It's in the name. So now that you know that, and we have this CDK cyclin complex, you have to realize something that you have to know is that, remember I said it's a kinase. It's going to phosphorylate something. So the thing that it's going to phosphorylate, and I just put a P here for meaning that's going to phosphorylate something, is going to phosphorylate something called retinoblastoma, or the retinoblastoma protein, the retinoblastoma gene. It doesn't matter. Basically, it's all pretty much the same thing. The gene basically produces the retinoblastoma protein. This name should sound familiar to you if you've been studying medicine for a while, because there's something called retinoblastoma, which is a disease that can show up in young children where they have this tumor that can happen in either one of their eyes or both of their eyes. And we won't get into too much details of that, but basically in that disease, the tumor is caused because there's something wrong with the retinoblastoma protein, which is in this pathway. So let's get back to how this pathway works. This is the way that I remember what retinoblastoma does without being phosphorylated. So you notice that it starts with an R. I want you to look at everything else that's in this pathway. E2F, CDK, cyclin, P53, P21, all of this stuff, nothing starts with an R. There's no R anywhere in here as far as what it starts with. That's going to help you remember, so I want you to remember, when you see R for retinoblastoma, I want the R to remind you of restrict. The R for restrict. Now there's a reason for that. In this pathway, when DNA, I'm going to ignore DNA damage for a while. Normally, retinoblastoma, it restricts the ability for E2F. So that's our next thing in this pathway. E2F is the final thing in the pathway. It restricts E2F's ability to work. Well, what does E2F do? E2F just simply causes the pathway to go on, the cell cycle. So E2F will literally bind into various proteins and activate various proteins and cause the pathway to go from G1 to S. Okay, and I'm going to write the rest of them here. So E2F, we want it to work if everything is okay in the cell, or the DNA of the cell. If everything is okay, we want E2F to be activated. But what the problem is going to come in when RB is not allowing it to be activated. Now, the only time RB will not allow it to be activated is when P53 detects that there's some sort of damage over here in the DNA. And I said a little bit of this pathway earlier, but now I'm going to walk through with all of these components and show you how everything works, and then I'll give you the rest of the mnemonic. So let's go back to the beginning. P53 has detected that there's some sort of damage in this particular cell's DNA. It is going to activate P21. Now, I don't have a clever way to memorize that it activates P21, so that's something you'll have to learn on your own. Just know that P53 detects the DNA, and that should be easy to remember because it is a tumor suppressor, so it's going to clearly suppress this pathway somehow, and we'll see how. It is going to activate P21. When P21 is activated, just remember that P21 is a CDK inhibitor. I said over here that CDK, when it binds to cyclin, it has to be bound to cyclin to work. When this complex is together, it will phosphorylate retinoblastoma protein. Think of it this way. When retinoblastoma has a bunch of phosphates, when it is phosphorylated, whether it's just 1 or 2, or that stuff's not important. Just know that when you phosphorylate retinoblastoma, it now struggles to bind to E2F. Just imagine the phosphates get in the way. Okay? So when retinoblastoma is phosphorylated, it now cannot bind to the E2F. What does that mean? That means that the E2F can now go ahead and continue on from G1 to S. But I've said earlier that in the case of DNA damage, P53 will activate P21, and P21 is an inhibitor of this kinase, of CDK cyclin complex. When you inhibit CDK cyclin, so when this is inhibited right here, you're not phosphorylating RB. And when RB is phosphorylated, its true function, remember it starts with an R, so remember restrict. Its true function to restrict will now be able to play in. How does it restrict it? Well, E2F is here. All RB is going to do is combine to E2F. Say this is E2F, this little rectangle. RB will just bind to this, and now E2F can no longer do its function. It's basically restricting its function. Okay, but when RB is phosphorylated, like say, let's go over here, when RB is phosphorylated, let's say there's a bunch of phosphates now on it, because what was it phosphorylated from? From the CDK, say in the case that P53 is not detecting any damage, nothing's wrong. Now this RB can no longer bind an E2F. The E2F is free to go on and cause the cell cycle checkpoint from G1 to S to be cleared, and then DNA replication, which happens in S phase, can occur. We can go to G2, and we can eventually then duplicate these cells. Okay, so I'm sorry if I didn't explain that well. Let me try it one more time, and I'll show you the last mnemonic. Let's go through this one more time, so you can even slow down the video if I'm talking too fast. We have a cell, and a cell has some sort of DNA damage. So in this spot, you notice there's some sort of weird DNA damage. P53, which is a tumor suppressor, detects that. So what is P53 going to do? It's going to activate P21. P21, you have to remember this, is a cyclin-dependent kinase inhibitor. Okay? So normally, cyclins, when cyclins are around, they will bind to the cyclin-dependent kinase. That's easy to remember. It's dependent on cyclins for it to work normally. And normally, they will phosphorylate RB. Normally, the cyclin-dependent kinase with cyclins present as a complex will phosphorylate RB. But in the case when P21 is activated from P53 because cell damage or DNA damage was noted, it will now inhibit, this entire complex is inhibited. Now we can no longer phosphorylate retinoblastoma protein. So what does that mean? Retinoblastoma no longer has all of these phosphates around it. So now it's a free molecule to go on and attach to E2F. And E2F is the final thing that's going to go on and allow the G1 to S to basically go from G1 to S, for it to go past the restriction point. Remember I said the R point is that checkpoint from G1 to S. So if now we've basically had DNA damage, and we have stopped the phosphorylation of retinoblastoma. So these aren't here. Now retinoblastoma is free because there's no sort of like spatial problems. It's able to easily attach to E2F. Now E2F, it shuts E2F down. Now here's the mnemonics to help you remember a lot of this stuff that gets really confusing. RB, I said that R stands in its normal function. It restricts. It restricts E2F in its normal function. So what does that mean? When it's not phosphorylated like crazy from CDK cyclin complex, in the case of P53, P21 noticing damage, when it's not phosphorylated, its normal function is to restrict this whole pathway. So you're stuck at G1, and that's to protect the cell from going on in a cell that, say, has damage or is faulty and that can cause problems later on. So R for restrict. R for restrict. It's the only thing that starts with an R, so this will help you to remember this. R basically restricts. But in the case of phosphorylation, when no damage is detected, it can't bind E2F, and that means the cell is healthy and E2F can function. Now, how do we remember what E2F does when it's not basically messed with? Just like I said, when RB is not messed with, it wants to go bind it wants to go bind E2F when it's not phosphorylated, and its function is to restrict. What about E2F? This is what I think about. E, The E and E2F, let's look back over here at our main little things we're talking about in the video. There's only one thing that starts with an E. There's not an E in any of these other units. So let's think of something that starts with an E that'll help us to remember what's going to be happening. And I think of entry, or like enter. Why? Because we're going to enter into S phase. G1 is entering into the S phase. So when E2F is free, when it's not being messed with by RB, because RB is phosphorylated like crazy, then E2F can function. Then we can have the actual function of E2F, and that is to allow the cell to enter from G1 into S through a series of complex processes, but you don't need to know it. So I'm going to go through the story one last time without stopping, and then I'll finish the video. So let's do this. We notice cell damage. Okay, P53 notices it, which is a tumor suppressor. It will activate P21. When P21 is activated, P21 acts as a cyclin-dependent kinase inhibitor. It's going to inhibit this complex of cyclin and CDK that is bound to each other. Normally, cyclin-CDK complex wants to phosphorylate RB. And that's in the case when no cell damage is present. When it phosphorylates RB, RB can no longer attach to E2F. There's just too many phosphates all around it. See all these phosphates? It can no longer attach to E2F. So that leaves E2F free to have its normal function, which is to allow the cell to go from G1 to the S phase to start DNA replication. But in the case of P21 being activated, it's shutting down CDK. Therefore, we're not phosphorylating RB. Now RB can have its true function, and I said it starts with an R. R stands for restricts. It will restrict E2F. And when E2F is restricted, it won't allow going from G1 to S. It shuts E2F down. But say RB was phosphorylated in the case of a healthy cell. That means that E2F will have its normal function. And how do we remember the normal function of E2F? Because it's the only thing that starts with an E, and E, then just think of entry into G1 into S phase from G1. Okay, so E for entry, R for restrict. Those are the functions when they're not messed with. RB can be messed with by phosphorylating it like crazy. E2F can be messed with, or its ability shut down, when RB binds to it. So I hope that simplifies the entire process. Let me clear all this and kind of give you an example of like sort of questions they may ask you. They'll basically say something along the lines of any of these molecules down here being shut down, not working, increasing, and then they'll say what will happen. For example, they'll say you've damaged a bunch of cells. Okay, you've damaged a bunch of cells. What will happen to this pathway? Well, if you damage a bunch of cells, just think common sense. You don't want those cells to go on and be produced or like duplicated, right? Because if you have a bunch of damaged cells, you don't want more damaged cells. They'll be produced incorrectly. So then that should help you remember. Then they'll say, well, what will happen to, let's say, the levels of RB in a damaged cells? Or will it be phosphorylated or dephosphorylated? So think about this. If you have a damaged cell, P53 is activated. It's going to activate P21. P21 is a cyclin-dependent kinase inhibitor. Now cyclin-dependent kinase cannot go and phosphorylate RB. Because remember that cyclin-dependent kinase normally it's a kinase. It wants to phosphorylate something, but this is shutting it down. It detects the damage. Because it can't be phosphorylated, RB is able to run rampant through the cell. And RB is free and no longer has all these phosphates around it, so RB can easily bind to E2F. When E2F is bound by RB, it's shutting down E2F. So E2F will not allow the progression of G1 to S. So you can see there's multiple ways they can ask this. They can say what will happen to the function of CDK or cyclin in case of damage, or in a healthy cell, what will retinoblastoma be phosphorylated or dephosphorylated? What will be the activity of E2F in a healthy cell or a damaged cell? So if you can remember the little mnemonics, RB, remember for restrict, it will restrict when it's not phosphorylated or not has a bunch of stuff around it. And then E2F stands for entry. It will allow entry into the S phase as long as RB doesn't interfere with it. If you can remember those little helpful mnemonics and think just common sense. CDK stands for cyclin-dependent kinase. It's telling you it's dependent on this cyclin, and the K for kinase means, think of what a kinase means. It's going to phosphorylate. You can basically talk your way through this whole pathway and figure it out. So if this video helped you in any sort of way, please like, subscribe, share the video. I will see you in another video next time. Bye, guys.